Thank you very much. Can everybody hear me? Okay. So very good afternoon. Today I'm going to speak about understanding the genetics of hair shape and what we inferred using the various mouse models. So before I go into, I would like to introduce you to some facts about the hair. So hair, as we know, is quite distinct part of our personality. It actually defines us in a way, and we have very different types of texture, color, or shape of hair existing even among the people sitting here. What is interesting is the hair shape is actually quite heritable, which means it gets passed on through generations. Oh, sorry. So what's interesting is that it's quite heritable. That means that it gets passed through generations. And if we actually kind of classify the world hair diversity, then we can broadly categorize into 4 main categories, which can be straight, wavy, curly, and very curly. Now, coming to the diversity among populations, if we see the African populations, then they are more have the kinky or the curly phenotype, whereas the Europeans are quite diverse, so they have straight, wavy, as well as curly. In case of East Asians, we see mostly the straight phenotype. So hair shape genetics and what we have learned till date. So it's a quantitative trait, and it's being determined by more than one gene. But what is very difficult, it's quite difficult to measure, so it doesn't come to a numerical value like any other phenotypic trait, for example, height or weight. And that's why, because it has many features, maybe curliness, maybe texture, maybe thickness, to count on. And that's why, even though the genome-wide have been quite an era since the last 15 years, we have had very few studies related to the hair shape. Until now, we know about 18 loci being contributing to the natural hair shape variation. And a recent meta-analysis data was being published. What is also interesting is that we have different genes contributing to the similar hair phenotype in different populations. So for example, we have the ectodysplacin A receptor gene, which contributes to straight hair in East Asians, whereas we have the trichotillin gene, which contributes to straight hair in Europeans. Some of these loci are actually positively selected, which means it's a very good point of study for the human evolutionary path, why they have been selected, what has been the selective phenotype. And also some of them are pleiotropic in the sense that it contributes to any other phenotype than the hair shape itself. But today's journey is on the PRSS53 gene, which is a gene coding for a serine protease. So it was first reported by a genome-wide association study, a collaborative study done from our group, in Adhikari et al. In Latin Americans. So we have the SNP RS111-50606, which actually leads to an amino acid substitution from glutamine to arginine. And if we see the 1,000 genomes data, then we can see that it's quite very evident in the East Asians, but not in the other world populations. We have also seen the evidence of positive selection. However, the selective trait and how it has been selected has not been investigated till date. So what this mutation actually does, from the evidence what has been learned so far, so bioinformatic analysis suggests that when we have, for example, the ancestral allele, then it actually, after the signal peptide, it introduces, we have the cleavage site here, but for example, if it turns to derived allele, then it introduces a proprotease, another cleavage site, which actually affects the processing and maybe the secretion of the enzyme. So our study design basically consisted of 3 main aspects. We were obviously interested in the phenotypic assessment of the PRSS53Q of 30R mice, but we were also interested in the knockout because the knockout wasn't studied till that, so we thought it would be a good reference to join in. We were interested in the expression of PRSS53, where it is expressed, and also how or the mechanism, if we can learn something about the mechanism of action of the gene. So these were the 2 mouse models that were generated. So we have the knockout here, and we have the point mutation mouse here, and these both were generated using CRISPR-Cas9 technology. So what are the phenotypes or the traits that we studied? We were interested, obviously, in the hair phenotype, given the human association, hair shape being the prominent one, but we were also interested in hair thickness and hair length. We were interested in the skin phenotypes as well because we had also an evidence of expression in the skin. So we actually measured the thickness of the epidermis, the dermis, and the fat layer. We were also interested in the glandular phenotypes, especially the sebaceous gland and the sweat glands, because we hypothesized that even the sebum acting as a layer on the hair shaft might contribute to the straighter hair. But due to time constraints, today I will be concentrating only on the two of the phenotypes which we found interesting, that is the hair shape and the skin epidermis. So what did we find in the knockout mice? So we found that the knockout mice had wavy coat and curly whiskers. I'm not sure if the wavy coat is evident, but certainly the curly whiskers will. And which made sense because in humans it was contributing to straight hair, so when we knocked off, then we got the curly whiskers. However, as science is evident, so we didn't get any observable phenotype, GWAS phenotype, in the point mutation. And that actually led to a more specific hair shape analysis for us. So one would argue, how would we use mouse model for our study? Because humans have curls, but mice have kinks, actually. And if we actually know about the 4 types of mice hair, which is present on the body, so we have the gourd and the awl hair, which are the straight hairs. We have auchin, which has 1 kink, and we have zigzag, which has 2 or more than 2 kinks. And actually, if a mutation actually contributes to straightness, then we would have more number of the gourd or the awl comparative to the auchin or the zigzag. So what we did was we took the body hair plucked down from bare skin from one single location of the mice, and we analyzed about 200 hairs per mice, and we categorized each hair as having 0, 1, or more than 2 kinks. And what we observed was we did find an effect of genotype on the hair shape, but interestingly, we didn't find in the 19-day-old or the 60-day-old mice, but we did find it in the 84-day-old mice. Coming to the skin phenotypes, so here on the left-hand side, you can find the H&E stained back skin. So we made the paraffin-embedded sections, and then we actually measured the epidermis, the dermis, and the fat layer. And we didn't find any significant differences in the dermis and the fat layer. However, in case of epidermal thickness, as the graph shows, we did find a difference among the wild types and the het and the homozygous variants. What you see is a representative data, and each data point represents a mice. So there are combined of 91 mice presented here. And we did find that the point mutation actually contributes to the increased epidermal thickness. And we did find the effect of the genotype. So next, we were interested in the expression of the PRSS53 gene, and for this, we used the in situ hybridization, and we used the modern method of RNA-scope to find out, and we did very many tissues, but where we found here, so on this side, you can find the negative control, which is the DAB-B, and here we can find very evident brown signals on the superbasal layer of the epidermis. And in this case, this is the dermal papilla, and we can find the very significant signals in the differentiation zone of the hair follicle, which actually congruence to our study, which we found in the hair or the skin phenotype. Then we were interested next in if we can know something about the mechanism of action of how PRSS53 acts. As you would remember that I introduced you to the EDR gene, so it contributes to actually straighter hair in East Asians and also being positively selected. So an obvious question to ask was, does PRSS53 interact with the EDR signaling at the cellular level? And for this, we conducted a dual luciferase assay by co-transfecting the PRSS53 variants together with the cDNA of the EDR and in the hex cells. And what we found was that if you can see, this is the empty vector, the wild type, and the Q30R, and here is the EDR, the wild type, and the PRSS53 ancestral, and the Q30R. Now, if we would sense an interaction of the PRSS53 with the ETR signaling, then we would expect a quite very increase in the signal, and whereas we didn't find it significant. So we thought that the PRSS53 might be influencing the hair shape via a different pathway, irrespective of the EDR. So coming to the conclusions, the fact that in the phenotype of curly whiskers and the wavy coat that we observed in the knockout mice actually attests the involvement of the gene in the hair shape. However, the effect of the Q30R variant was only seen in one particular age point and not in the others. And we also come forward with a novel phenotype, that is the effect of increased epidermal thickness in mice, which can now be tested back in humans, and that's the whole point of conducting a mice model study. And it also told us that it contributes to phenotypes beyond hair shape. And since we did not observe an interaction with EDR signaling, it suggests that PRSS53 and EDR might be influencing the hair shape through independent molecular pathways. What we are next interested in, and would be interesting for us to know, contributing to the epidermal thickness. So we actually are now going to ask the question if the thickening of the epidermis it's actually whole of the epidermis layer, or it is one of the 4 subparts of the epidermal layers. And for that, we are using immunostaining and for these epidermal markers. What we are also interested in is the hair strength measurements, because from our data, it appears that it may be involved in the keratinization, and so we would be conducting some hair strength experiments together with the Heriot-Watt University. And another mutation, which is just exactly 10 base pair away from what we have been studying, is the PRSS53 G34S. The interesting thing about it is it's selected in South Asians. So we have one mutation which is selected in East Asians and another being selected in South Asians. And this we are going to do the study in collaboration with Wellcome Sanger Institute. So I would like to acknowledge Dennis, who has been supervising and guiding me through the study, and also Professor Ian Jackson, who is here, for being a mentor for all the studies, my fellow lab mates, and Marie Curie Individual Fellowship for funding the study. Thank you very much.
Thank you very much. Thank you very much indeed. Any queries or comments? Right at the back.
Yeah. Do you know if there's a difference in cross-section shape for different hair types?
I'm sorry, I can't hear you.
The shape of the cross-section of different hair types.
Okay,
do you know if there's a difference there?
So there has been some studies which show that the shape of cross-sections are different across the different hair shapes, but it has not been very consistent. So they put forward the idea, but it has not been very consistent. We did not do any study on cross-section that would involve basically SEM or the TEM analysis.
Hi there. I might have missed it at the beginning, but is there any evidence for natural selection on these loci across populations?
Yeah, so EDR has been positively selected. Also, the PRSS53, which I'm being studying, is also positively selected in East Asians. So yes, there is evidence of positive selection, and that's why the bigger question is what is the selective phenotype? And so through these mice model studies, we can actually go through, except for hair shape, what are the other phenotypes, and if any of these phenotypes can actually be a reason for positive selection in humans, and that would have led to the high frequency in the population.
One of the phenotypes you picked up was which have a level of skin it was, or thick skin or thin skin.
Yeah.
Is that something you think is more general, or?
Well, this is what we got from our results. Possibly doing it back in humans would tell if the effect is being consistent. So there can be different reasons for being, and there are measures to do the epidermal thickness being measured in human skin. We have a non-invasive and invasive approaches to do that.
Any other comments or questions? I think we're just about a good time. So, well, thank you very much. And that's thank all the speakers.
I'd like to thank all the speakers, and for a very entertaining morning. Just a couple of announcements. Lunch is going to be available downstairs. There are posters to see, and I've asked the poster presenters to be there in the sort of second half of the lunch break so that you'd have an opportunity to talk to them and give them encouragement. And we'll get back in an hour's time for the afternoon session, which I'm sure you're all looking forward to.